Here we can see how the DNA is getting replication. It's a very interesting thing. At the time of replication, two strands of DNA separate. Here you can see these are the two strands of DNA. We told it they are complementary to each other. The two strands you can see they are separating. Okay, this is one strand, this is another strand. Each strand acts as a template for the formation of new strand. So each one, this is one, this is the second one, each act as a template for the formation of a new strand. A new strand is constructed on each old strand by complementary base pairing. So here you can see how it is happening. Now you can see here thymine. So next to that adenine will form. Here guanine, cytosine will come. Here cytosine, guanine will come. Again adenine, thymine will form. So a new strand is constructed on each old strand by complementary base pairing. You can see here there is a formation of new strand. Clear? So this is the old one, this is new one. Here also this is the old one, this is new one. So they will be doing the complementary base pairing. Hence, two exactly identical double-stranded DNA molecules are formed. So what will happen? Two exactly identical double-stranded DNA molecules are formed. How it was this a parent one? So you can see same like now here we are getting two. Clear? In each new DNA molecule, one strand is old while the other is newly formed. So this is old one, this is newly formed. Here also this is old one, this is newly formed. Hence, Watson and Crick described this method as semi-conservative replication. So because of this one is old, one is new. Watson and Crick gave one name. What name they given? They have given semi-conservative replication. So this is the meaning of semi-conservative method of DNA replication. Clear, children? So this is the parent DNA. I told there are two strands. First it will separate. Here there is a complementary base pairing. Along with that one base pair will join and they getting new strand. So this is the old strand, this is the new one. Same like here, this is the old one, this is the new strand. So here another same thing you can see here. A semi-conservative nature of DNA, Matthew Michelson and Franklin Starr. Matthew Michelson and Franklin Starr in 1958 performed experiments in E. coli to prove that DNA replication is semi-conservative. So there are two scientists, Matthew Michelson and Franklin Starr in 1958 performed experiments on E. coli, Escherichia coli, it's a bacteria, to prove that DNA replication is semi-conservative. Let us see what they did. Yes, here you can see semi-conservative nature of DNA, Matthew Michelson and Franklin Starr. What they did, they grew E. coli. E. coli, Escherichia, full form is, scientific name it is Escherichia coli. It is a bacteria on 15 ammonium chloride. Here it is one of the heavy isotope of nitrogen. Nitrogen 15 is one of the heavy isotope of nitrogen. So 15 ammonium chloride culture medium. Nitrogen 15 is the heavy isotope of nitrogen. Both strands of DNA have, so DNA we told it's a double stranded. Both strands of DNA have nitrogen 15, N15, N15. These bacteria are shifted to 14 ammonium chloride culture medium. So before it was 15 ammonium chloride, but now here it is 14 ammonium chloride. Here this is nitrogen 15. It is one of the isotope of nitrogen. Here now it is nitrogen 14. Okay, another isotope of nitrogen. This is heavier one. This is lighter one. So these bacteria are shifted to 14 ammonium chloride culture medium. DNA extracted subjected to CSCl, it means that is cesium chloride. CSCl, full form it is cesium chloride. DNA extracted subjected to CSCl density gradient centrifugations. Okay, centrifuge means usually it is for the separation of the virus particles. We learned before that is we are using this centrifugation. So DNA extracted subjected to CSCL, that is cesium chloride density gradient centrifugation. Then after that, they will be getting hybrid or F1 generation. This hybrid or F1 generation, it will be intermediate type of DNA, that is N15 and N14. So this is the F1 generation or hybrid. They will be getting intermediate type of DNA. It's not like parent like, intermediate type of DNA N15 and 14. Nitrogen 15 and nitrogen 14. These are the isotopes of nitrogen. After next generation, then they came to the next F2 generation. Equal amount of light DNA, that is N14 and 14, and hybrid DNA N15 and 14 are formed. So when they came to the next F2 generation, they'll be getting both the mix. When they are getting light DNA N14 and 14 and hybrid DNA N15, nitrogen 15 and nitrogen 14 are formed. Clear? So I can show you here. Yeah. Look at this. Here, this is the parent one. You can see nitrogen 15 DNA, dark color. This is generation 1. Here they will be getting both. You can see here light color and dark color. Here also you can see light and dark color. So the light colors, they are, see here, N15 DNA, that is a dark one, N14 DNA, that is the light one. So both they are getting. So that is a generation 1. Hybrid is both mix intermediate N15 DNA, N14, nitrogen 15 and nitrogen 14 DNA. Again came to the second generation. Here you can see all the mix. The light color, that is nitrogen 14 DNA. Here you can see dark and light both mix. The dark color, it is nitrogen 15 DNA, heavier one. Again you can see the light colors, that is nitrogen 14 DNA. Again you see here light color, that is nitrogen 14 DNA. So dark colors nitrogen 15, light colors nitrogen 14. Okay, that is the isotope of nitrogen. Again you come down here, you can see after centrifugation, here you can see N15, N15, that is heavy, N14, N15, that is hybrid, nitrogen 14, that is light DNA, nitrogen 14, nitrogen 15, that is hybrid. So in the second generation, they will be getting all the mix. It means heavier 15, that is heavy, 14, that is light, 14, that is intermediate, that is the hybrid form. So in the second generation, they will be getting all different types. Clear? So same thing is given again, semi-conservative replication in this parent DNA, N15, N15. In the first generation, they will be getting the hybrid, both mix, N15, N14, N15, N14. But in the second generation, they will be getting all the mix they will be getting. So that is semi-conservative nature of DNA. It is proved by two scientists, Matthew Michelson and Franklin Stahl in 1958. So what is the mechanism of DNA replication? What is the main mechanism of DNA replication? The process of DNA replication takes place by number of substance, enzymes, and proteins. So for replication, we need three things. Which are the three main things? Number of substance, second enzymes we want, and proteins. These are the three things taking place during the time of DNA replication. Which is a substance? Deoxyribonucleotide. Clear? Substance is deoxyribonucleotide because it's a DNA. If it is RNA, ribonucleotide. If it is a DNA, deoxyribonucleotide. Enzymes, DNA helicase, DNA polymerase 321, RNA primase, DNA ligase. Protein, SSB. What is SSB? Single strand binding protein. Single strand binding protein. Clear? So I'll repeat once more. Substance is deoxyribonucleotides. Enzymes 4 types: DNA helicase, DNA polymerase 321, RNA primase, DNA ligase. Protein, SSB, that is single strand binding protein. SSB is single strand binding protein. So all this you can see here, template strand, these two bluish color, these are the template strand. This is called a replication fork. I'll tell you what it is, replication fork. Then DNA polymerase, the green color. DNA polymerase, here also you can see greenish color, that is DNA polymerase. Yellowish color, that is the DNA ligase. Then Okazaki fragments. I'll teach now what is that Okazaki fragments. Then here you can see lagging strand, and another one you can see leading strand. So this is leading strand. This one is lagging strand. Here you can see Okazaki fragments, DNA ligase, DNA polymerase. Here also you can see DNA polymerase, replication fork, and template strands. I'll explain each one. The same thing here also they given leading strand template. This red color leading strand, lagging strand. This one is Okazaki fragments, like you can see there is Okazaki fragments. After that bluish color, that is the lagging strand. So this is lagging strand, this is leading strand. Clear? Then DNA polymerase third elongates the lagging strand. DNA polymerase makes primer. DNA polymerase third elongates the leading strand. Helicase unwinds the double helix. This is a parent DNA. Single-stranded DNA binding proteins makes the templates available to RNA primase and DNA polymerase 3. So I'll explain to you first, then I'll come back here again. So mechanism of replication starts at a specific point of DNA molecule called point of array. So mechanism of replication, where it start at a specific point of DNA molecule called point of array. Okay, so here the replication it starts. This point is known as point of array. At origin, DNA strand unwinds by breaking hydrogen bonds. So how it is unwinding? It unwinds by breaking hydrogen bonds. This takes place with the help of an enzyme, DNA helicase. It unwinding part. I told how it unwinds by breaking the hydrogen bonds. It takes place with the help of an enzyme, DNA helicase. I'll come back and I'll show you here. See, helicase. Helicase, this is a DNA. Helicase unwinds the double helix. Okay, so this is double helix. Now it becoming separate. Here you can see the separate. Single. Who will help? Helicase unwinds the double helix. At the point separation, it appears like a fork or a Y shape. It is called replication fork. At the point of separation, it appears like a fork. You know what is fork? Or a Y shape. It is called replication fork. Look at here. See, it is getting Y shape or is getting fork-like shape. That is called replication fork. Got it? The next is each old DNA strand acts as a template for the synthesis of new strand. So each old strand, before it was double, now it became single. Each old DNA strand acts as a template for the synthesis of new strand. SSB protein, we told there is one protein that is called SSB protein, attaches to unwinded strand and prevents rejoining. So what is the function of this protein? This protein attaches to unwinded strand and prevents rejoining. Otherwise, again, it will go and join. Sometime it may happen. So to prevent that and rejoining, this protein attaches to unwinded strand. Look at here. See, you can see here black color. Here also you can see the black color portion. This is the protein. So single-stranded DNA blinding proteins make the templates available to RNA primase and DNA polymerase third without rejoining. Okay, so this black color is the protein. Here also you can see black color. It is attached to this single strand. Why it is attached? Otherwise, again, these two bluish color, you can see this strand, it will come and rejoin sometime. To avoid that, the proteins attached to this strand. Single-stranded DNA blinding proteins make the templates available to RNA polymerase and DNA polymerase third. The synthesis of new DNA strand on old strand takes place by an enzyme DNA polymerase third. It adds new nucleotides through complementary base pairing. So the synthesis of new DNA strand on old strand takes place with the help of which enzyme? By an enzyme DNA polymerase third. It adds new nucleotides through complementary base pairing. Okay, you can see here, see, DNA polymerase third elongates the leading strand. Here also you can see DNA polymerase third elongates the lagging strand. I'll tell you what is the leading strand and lagging strand. Before that, you see this DNA polymerase third elongates the leading strand. DNA polymerase third elongates the lagging strand. DNA polymerase third always synthesis new strand in 5 to 3 direction. So what is function of this DNA polymerase? It always synthesis new strand in 5 to 3 or 3 to 5 direction. Then now let us see what is leading strand, what is lagging strand, what is Okazaki. The synthesis of new strand for the parent template strand having 3 to 5 end is continuous. This strand is called leading strand. The synthesis of new strand for the parent template strand having 5 to 3 end is discontinuous. It forms by small segments of DNA called Okazaki fragments. This strand is called lagging strand. During the formation of Okazaki fragment, RNA primase first synthesize RNA primer. So I'll show you what is leading strand. The synthesis of new strand for the parent template strand having 3 to 5 end is continuous. This strand is called a leading strand. I'll show you that. See, look at here. This is leading strand. This is bluish color. Okay. So one is old one. Sorry, this is a red color. So this is the old strand. Here you can see there is one 3 to 5 new strand is coming. Continuous it is coming. 3 to 5 new strand is coming. This is called a leading strand. But here you see 5 to 3, one strand is coming, but it's not continuous. Here you see red color, it's continuous, you can see. Here you can see red, green, red, green. It's coming like small fragments of DNA. Okay. This fragment is called Okazaki fragment. The fragments are called Okazaki fragments, and the strand is called lagging strand. Clear? So this red is leading strand. It is from 3 to 5. Then here this is lagging strand, that is 5 to 3. But what is Okazaki fragments? Here you see again small fragments like DNA. You can see these are known as Okazaki. It is not continuous. These are discontinuous. This is a continuous one, but this is the discontinuous. So that is called each fragment, small piece. It is known as Okazaki fragment. Clear? So synthesis of new strand for the parent template strand having 3 to 5 end is continuous. This strand is leading strand. Second one, the synthesis of new strand for the parent template strand having 5 to 3 end is discontinuous. It forms by small segments of DNA. It is called Okazaki fragments. This strand is called lagging strand. During the formation of Okazaki fragment, RNA primase first synthesis RNA primer. That also I can show you here. See, this one RNA primase. What they make? They make primer. Okay, so these all enzymes. RNA primase, they make primer here. During the formation of Okazaki fragment, RNA primase first synthesis RNA primer. Then DNA polymerase third continues the synthesis of Okazaki fragments in 5 to 3 direction. At the end, RNA primer are replaced by deoxyribonucleotides with the help of enzyme DNA polymerase 1. The Okazaki fragments join together with the help of an enzyme DNA ligase. DNA polymerase 2 once again checks and repairs the errors occurred in new strand.